Hello, everybody. In this opportunity, we are going to show our final project. The title of our final project is the development of a sensitive and specific wholesale biosensor for the fluorometric and colorimetric detection of bioavailable and cadmium in different substrates, presented by Carlos Castro and Miguel Grandes. Here we have the main table of contents with the topics that we are talking about in this opportunity. To do our proposal investigation, we have to ourselves this question: is it feasible to develop of a sensitive and specific wholesale biosensor for the fluorometric and colorimetric detection of bioavailable cadmium in different substrates? Okay, before to talk about the cadmium, we need to know how the cadmium ends on the environment. Okay, to talk about it's necessary to talk about the industrialization. The rapid growth of the industrialization has caused the presence of chemicals, including heavy metals, in the environment, like the cadmium. The cadmium could be present in many substrates, like the water, soils, in another kind of substrates, and the air. It can get into the human body in three ways: inhalation, dermal absorption, and ingestion. The cadmium could cause many problems on the human health. Could have problems with the immune system, problems on the lungs, kidneys, testes, brain, cardiovascular system, bones, and liver. In view of this problem, there has been developed many technologies to monitoring this problem, the cadmium in the environment. The problem is that these technologies are not cheap, are too expensive, are not portable, and need a long time of preparation samples to do the analysis. So nowadays has been developed new technologies like the biosensors. We are focused in two kinds of biosensors: the enzyme biosensors and the wholesale-based biosensor as a micro-nano biosensor. Because these biosensors are better because are cheaper, are more portable, and are more faster to do the analysis. You can do the analysis in vitro, in situ. So now we have the interaction between the bacteria and the cadmium. We can see in the first part, and how the cadmium environment could be absorbed by this bacterial cell wall. Then for the MIT metal and the MIT system, the cadmium will get into the cell, and for gene, the CADCA operon, the cadmium will be a flux out the cell. We can see another mechanism like the cadmium sequestration by the protein SMT. This is our mechanism to defense of the cell to the stress caused by the cadmium. Now we can see how the cadmium, how is the function of the cadmium. The cadmium have three main elements: the analyte, the reporter gene, and the product. The analyte in this case will be cadmium. The reporter gene will be the gene who will give a response when detect cadmium. This response could be a protein, a luciferase, and another kinds of response. And finally, this metabolite will have a response like a fluorescence, bioluminescence, colorimetry, and another response. We have done this hypothesis. The developed whole cell biosensor is sensitive and specific for the fluorometric and colorimetric detection of bioavailable cadmium in different substrates. Our objective is to develop a sensitive and specific whole-cell biosensor for the fluorometric and colorimetric detection of bioavailable cadmium in different substrates.
In the experimental procedures, we are going to work with E. coli strain top 10 on the plasmid. Next. Those materials are for the stock solution and the culture media. By the way, we are going to use kits as plasmid purification and sequencing kits. Those are materials for the gels in electrophoresis process. This is the equipment for genetic transformation and sequencing. For fluorometric and colorimetric analysis, the spectrophotometer and the multi-mode microplate reader. And for the bacteria culture, we have the shaker and the incubator. And finally, for electrophoresis, we have this electrophoresis system and the gel viewer. Then we have the assembly of a genetic construct for biosensing of bioavailable cadmium. The construct has two components. One that serves to detect the CAD-C gen signal, which consists of a promoter and also has a reporter component. But here, the reporter is formed by two fused proteins. The first one is the gene that encodes for the green fluorescent protein that superfolds in GFP. Its structure is not altered when it binds to another protein. That's why this variant of the green fluorescent protein was chosen, because this one possesses this particularity. It was mutated in such a way that it can keep its structure intact when it has fused it to another protein. This fusion protein is formed by the green fluorescent protein that it's bound to a blue chromoprotein. These are its two components. When this system is expressed in the presence of cadmium, as we can see in this slide, it will be expressed as a fusion protein that will have the green fluorescent in this part and the blue chromoprotein, so that it will be possible to detect the signal in the presence of cadmium by fluorescence, thanks to the green fluorescent protein, or by colorimetry, where the colonies genetically transformed with this spectrum will develop a bluish pigmentation. The production of the protein occurs simultaneously, thanks to its peptide linker keeping both proteins with its structure. This is known as a chimeric protein. This plasmid has the promoter of CAD-C. That promoter is the system for detecting the presence of cadmium in the environment. The component that it will give the colorimetric signal. The biosensor has two genetic components. One is the cadmium detection system, as I mentioned, and the other is the reporting component that gives the fluorescent signal. It also has the ampicillin resistance gene, and therefore this bacteria must be grown in ampicillin-containing culture media. It also has its origin of replication. Once a bacterium is transformed with this plasmid, the bacterium will begin to replicate more copies of this recombinant plasmid. This plasmid is of the high copy number replication category. This origin type can have like 500 copies of plasmid for each bacterium. That is why it was selected. This system is more sensitive and has a higher detection in high and low concentration of cadmium. So this is increasing the detection range of cadmium. Next. We can see in this slide the specificity test. And we are going to see different heavy metals that are going to interact with our system. Next. And in this case, we are going to look about a sensitivity test, in which one we are going to see the different concentration of cadmium in this experiment. So as we can see, the biosensor have a lot of uses. So for example, we can use with water, with biological tissues, with blood, with vegetable tissues, and have a lot of different uses. So we hope to have this biosensor done with a year of duration. This is a bibliographic reference.